Now we're going to talk a little bit about MR. So, what does MR stand for? Is it magnetic resonance? Is it material recovery? Is it metal recovery? Right. MRI stands for magnetic resonance. Even though we're going to have a whole explanation of how MR happens, we need to have kind of a basic understanding of what goes on with MR. So, what happens with MR is that you have a huge, very, very, very potent magnet. And the magnet that we're using is based, the way we measure the magnetism is in something we call Tesla. To just give you an idea of what a Tesla is, one Tesla is around 10,000 Gauss. And in your daily living, I'm sure you've seen a compass, and you can move the needle of a compass with the Earth's magnetism. The Earth's magnetism is half a Gauss. So when you're talking about one Tesla, you're talking about 20,000 Gauss, I'm sorry, 20,000 times the magnetism of the Earth. If you come to the hospital and you're going to check out some of our magnets, we have a bunch of 3 Tesla magnets. So when you're talking about a 3 Tesla magnet, how many times the magnetic field of the Earth is that? 60 times, 600 times, 6,000 times, or 60,000 times? Correct. 60,000 times the Earth's magnetism. That's how strong the magnet's field is when you put patients in a 3 Tesla magnet, which is, I think, very cool. It's a very interesting number to know. So what happens with MR is just a phenomenon that you transfer energy in the shape of radio frequencies into atoms that are all aligned by a big magnet. And we're going to review this again, but we're going to do a very simple demonstration of how that works. So you first pick the most frequent atom that you have in your body. So imagine which is the atom which is the most frequent that you have in your body. Which one is it? Would you say carbon? Would you say it's oxygen? You say it's hydrogen. Hydrogen. Your body is made up roughly 60% from water. And just a water molecule has 2 hydrogen atoms. But most of all the atoms that you have in your body have some sort of hydrogen. So you pick hydrogen. And by the way, hydrogen is a fantastic choice because it's the only atom that when you look at the nucleus, it does not have a neutron. It's proton. So when you put all of these hydrogen atoms into this huge magnetic fields, they will all align. And then they will all start have kind of a translation motion, like the Earth has a translation motion. And we call that the Larmor frequency. They do it in a frequency. And if you give them or transfer a radio frequency in the same frequency, you can tilt their magnetic vectors. So they transfer and they absorb that energy. Once you remove that, they're gonna release the energy, and you can detect it, and that's MRI. So you have a bunch of hydrogen atoms that are dancing together with the same frequency, the same rhythm, in the same vector direction, which is the direction of the magnet. You transfer some energy, they absorb it, and when you release the energy, you stop giving them the energy. They'll stand up back to their magnetic field, but energy is a constant, so they have to release that energy. And all you have to do is to have some sort of detector and tweak the atoms a little bit, and that's what MRI is all about. And I will only get to that today because we will definitely go a little bit deeper into that when we do the basics of MR physics. So if you understand that, when you do MRIs, we'll also have grayscales. But the grayscales that you're going to have in an MRI are not based on the density of the structure of the molecule that you're crossing x-rays. That's not it. The differences between them is how you tweak the magnetic radio frequencies for it to, when it releases the energy, how can you detect the energy? And the same way you have a TV set and you can change channels, you're using the same TV set and you're just receiving different channels in the same set. With an MRI, you can just change and tweak the way you do the experiment to receive sometimes water being bright, sometimes fat being bright, sometimes both being bright, sometimes not. So that's why in MRI, different than CT, you only have this big blob of information that you can display. In MRI, depending on how you tweak it, you're going to have a different response from the hydrogen atoms. And that's what we call our magnetic resonance sequences. And there are two main sequences that we use, T1 and T2. Remember that. Since we are not looking at densities, how the X-rays cross, but the intensity of signal is released by the atom. When things are bright on MRI, we call them hyper intense. When things are dark, that means that it was having very low signal. We call it hypo intense. You see the difference? Do you remember how black was on CT? Hypo, dense. How was bright on CT? Hyper dense. So when we talk about whites and dark grayscale, in MR you utilize the words intensities. So forget about the grayscale that we used on CT. In MRI, you do not use it, and it depends on which sequence you're looking at. So these are the two main sequences that we use on MRI. We call them T1 and T2. And there's a couple of rules, and if you know these two rules, you're always going to get this right. The first rule is only on T1 weighted images, only will the white matter be white. We already went through where the white matter was, right? We said the white matter was all of this region of the brain, which was not the cortex or the gray matter of the basal ganglia. So if you see it carefully, the white matter in this case is white. On the contrary, if you look at the image below, the white matter is always dark here. So just by looking at the white matter, you know if a sequence is weighted on T1 or on T2. So let's go this again. This is a T1, this is a T2. After you know that, remember the second rule. Only on T2 weighted images, will the water be bright. You will not have bright water on T1, but only on T2 is it going to be bright. Why is it the second corollary? Because there could be a chance that you can tweak a sequence to have a T2 with dark water. There could be the chance. So that's not the first one you use. You first look at the white matter, decide if it's a T1 or a T2. And if but if you see any sequence that has bright water, you know for sure it's a T2. We're gonna see why that is important. So the first thing you have to check is the bone white? So you look at the bone and see if the bone is white. If the bone is white, then it's going to be a CT. Only on CT is bone white. If the bone is dark, you have to be really careful to know where the bone is. I'm going to show you why. Then you know it's going to be an MRI. Then you go to the white matter. If the white matter is bright, that's the first corollary of MR. You know it's a T1. If it's dark, it's a T2. So just with these rules, with two little rules, you can do all the differentiation between a CT and an MR T1 and T2. Two rules. You don't have to go further than that. And a lot of the times when you get in the real world a case, you really need to know if you're looking at a CT or an MRI. Most important, what if you're in a board and they're asking you a question? You need to know what type of sequence or what type of study you're looking at. So this should help you guys a little bit with that. So this is one of the first questions I ask in this lecture. So you have these four, and you have to do the analysis, and you have to tell me which one of them is a CT, which one is an MR. So let's go to the first top one, which is A. The first thing we're going to ask ourselves is, how is the bone? So the bone is here. Is the bone white or dark? It's white. Let's go to the next one. In the next one, you can look at the bone. The bone is this dark part here. That's the bone. So it's dark. What about this one? It's dark. What about in this one? It's dark. So which one is the CT? A. You have to be able to do that differentiation, because if you look carefully, you're going to see that even though this is the bone, when you go to the other sequences, you can see that even though this one really looks bright right around there, you can see that this is bright. It's very different than where the bone is. Why? Because the scalp is the one that you're looking and seeing bright. So don't get confused between scalp and bone. Let's see in the next example if we get it better. But that was our CT. Now we have a new sequence that got invited into the question. So which one of these sequences is a T1? First of all, it has to be an MR. We already knew that this one has bright bone, so this one is not. So it has to be one of these, and one of these has to have bright white matter. So let's go to image B. Is the white matter bright or dark? Bright, correct. Let's go to image C. Is the white matter white or dark? It's dark. Let's go to image D. Is the white matter white or dark? It's dark. So it's easy to have an answer. So the answer is going to be B. B is the T1. Very well. So now we're going to introduce a new concept. So these three are MRIs. The first thing is we have to define which ones are T1 and which ones are T2. So let's go to this first one. Is the white matter white or is it dark? It is white. So that one is a T1. But then we have these two right here. And here's a trick. Both of them have dark white matter. That doesn't make any sense. So I had told you that if it's white matter is bright, it's a T1. If the white matter is dark, it's a T2. So we have T1 and two T2s. I had also told you that if the water was bright, it's a slam dunk T2. You don't have to do anything else. But then we ended up with this sequence, which has a T2. You know, it's a T2. White matter is dark. But then the ventricles are dark. What is going on with that? That doesn't make any sense. So I'm going to introduce you to the third main sequence that we use on MRI, which is a T2 where we saturate the fluid. And we call that fluid inverted recovery sequence, or FLAIR. Correct? So FLAIR is a T2 where you saturate the ventricular fluid, kind of. Why is it important? Because if there's disease like edema, it will not be saturated. It's going to remain bright. So the brightness of the ventricles or of the CSF around the brain will not be disturbing my interpretation of the image. So T2 is really important in both T2, regular T2, we're going to call it regular for now, and T2 with inversion recovery of the fluid, which we call flare. So now we should be able to differentiate CT from T1, T2, and flare. And that is the core of how we do neuroimaging. So let's test ourselves. One of the very important things is that we are able, with MRI, to do direct sagittals, coronals, axials, or oblique views because they just depend on how I tweak the magnet. So let's start first with the planes. You know that there are three anatomical planes. You have a plane that goes through the axis of the body. We call that axial. You have one that divides the body into anterior and posterior, which is called coronal. And then one that divides through the midline into symmetric halves, left and right, called sagittal, or slightly offset, which we call parasagittal. So which one of these is a sagittal view of the brain? Is it A, B, C, or D? Of course it's B. That's a sagittal view of the brain. You're going to need these planes and be very careful with the planes as you describe things when you're doing this course. There you go. That's your sagittal view. So let's conclude this first part of the course. Today, we were able to look at CTs and MRIs. We said that CTs are cross-sectional images that are based on X-rays. We also said that CTs, since they have X-rays, they use ionizing radiation. That ionizing radiation is very important to us, and we're going to have some questions on that and explain why. Because the high frequency of these photons can affect even the DNA. Another thing that we reviewed today is how you see the differences between CT and MRI in images. And the first thing we looked for was the bone, that if the bone was right, we were going to call that a CT. That you can have cross-sectional images of MRI by putting the body in a huge magnetic field. We reviewed that it was measured in Tesla, and that 1 Tesla is approximately 10,000 Gauss, and that the Earth's magnetic field is 0.5 Gauss. That MRI uses magnetism, so there's no ionic radiation involved. So it's much safer. That you can, in MRI, you can get different sequences, and the main sequence that we look for first is if it's a T1 or not. And the way to do that is look at the white matter. If it's bright, you know it's a T1. If it's dark, it's going to be a T2. We also saw that water is only bright on T2, but that we have a special sequence, a trick sequence that we use, which is a T2 where we can saturate the CSF, which we call the fluid attenuated inverted recovery sequence, or flare. And those are the main sequences. And the reason we do different T2s is because typically disease tends to have higher water content, so it's going to be bright on T2 and flare. So that is the end of our first lecture. Thank you for being here.